Hello, this is Dr. Billis. We're going to go over some lab highlights leading up to your lab exam for chapters 19, 20, and 21, which will be the blood, the heart, and your blood vessels. Okay, so looking at this picture, what you're seeing here are red blood cells, and you're seeing a white blood cell. Be able to identify the white blood cell as seen in this photo. I'll give you a hint. It's granulated, multi-lobed, a multi-lobed nucleus, and it's stained orange. You'll typically see this associated with hay fever, allergies, is what I'm trying to say, and possibly parasites. Okay, so that's your first photo. Your next photo, you have, again, red blood cells with a white blood cell. Key here, as far as identification, is going to be it's granulated, very much granulated, and it's a white blood cell. Okay, your next picture, if you look closely, you can see one yellow arrow and two green arrows. The two green arrows indicate you're looking at the same cell. And what you're seeing is a lot of red blood cells, and then you've got three white blood cells. Be able to identify those white blood cells. And then you've got an arrow also pointing at a little speck in this picture. Be able to identify what that little speck is. Hint, think coagulation. Okay. So be able to identify the white blood cells and also know, as I may ask you the function, what their functions are. Okay, next picture. Be able to identify this white blood cell. Single white blood cell amongst many red blood cells. I think a key here is, as far as identifying this white blood cell, notice that it's pretty much the same size as the red blood cells. Okay, next photo. Identify a white blood cell, or be able to tell me about the function. You see a white blood cell here that is much larger than the surrounding red blood cells. That is your clue. Okay, your next picture photo, we're looking at blood typing. We see a tray that has been filled with blood, and we're doing a blood typing match. What I want you to take away from this is that there is a reaction in A, and there is a reaction in RH, but there is no reaction in B. So this picture allows you to see clearly what a reaction looks like versus a non-reaction. Okay, so when it comes to blood typing, be able to tell me what the antigens are, as well as what the antibodies are that cause it to react. In this next photo, This is a chart that is used to measure formed elements as well as plasma. What did we call this lab experiment? It was a hematocrit. And can you tell me what the hematocrit looks at? Okay, I'll give you a second to think about it. Okay, your answer is packed cell volume. It's, well, I don't know if I left it in there so you could see it or not, but it's packed cell volume. We're spinning, spinning the blood down in a capillary tube, which is the little tube that you see in the chart. And we're left with the blood spinning down towards the bottom, and the bottom has been packed with clay, and we have plasma on top. Be able to tell me what the blood at the bottom, which is colored, it's red versus the clear plasma. Tell me what name is given to it and what is inside, what three elements make up the red portion. Be able to tell me what elements make up the plasma, what it's mostly made up of, and secondarily, if there are proteins in it, what is the most abundant protein in the plasma? Okay, so what this chart is used for is if you bottom out, zero out the bottom of the red, if you will roll it over to the left of the picture and put that on 0, then your plasma is going to be above your 100 mark. So to get a reading of what you're looking for, what you do is you take and you roll that capillary tube out or over until the top of that capillary tube is touching the top of your chart. Now when you do that, and you got the bottom lined up, and now the top is lined up with the 100 on the chart, you can take a simple reading of where that red is falling on the chart. And that will be your hematocrit reading. And an average reading for an adult would be able to tell me that. Your next photo, if you look at that closely, you'll be able to see 3 or 4, let's see, 1, 2, 3, 4, well, let's make it 3 green arrows. It's kind of hard to see, but the arrows are there. And they're pointing to lines which are lighter. Number 1, be able to tell me what type of muscle this is, and be able to tell me what those striations are that those arrows are pointing to. Okay. While you're at it, tell me how many types of muscle there are in the body. Well, I'll help you. 3, skeletal muscle, smooth muscle, and you can tell me the third one. Okay, in the next picture, what you're looking at is a picture looking from superior straight down at the base of the heart. Base of the heart are where the vessels are at. Now, you're not going to find this picture anywhere in your textbook. You're not going to find it in your lab book. You're not going to find it in any of your notes on Canvas. But you will find this picture to be able to study it on Google. So if you Google vessels of the heart, superior view, you'll probably be able to find it. So be able to identify the various valves or vessels on the base of this picture of the heart with this picture. We're looking at a picture of the heart. And we've opened the heart up, and we're looking at many different things on the inside of the heart. I'll be able to identify the right and left ventricles, the interventricular septum, pectinate muscles, atriums, the ligaments that hold the valves, helps the valves close, the muscles attached to those ligaments. So basically know the anatomy of the inside of the heart. Okay, continuing with the anatomy of the inside of the heart. As a fetus, there is a hole between the atrium. Be able to identify that hole and provide its name. Also be able to identify that there's a ligament that holds the pulmonary trunk to the aorta. Be able to identify that ligament by name. Also be able to identify some of the major vessels coming off the heart, such as the aorta, the superior and inferior vena cava, as well as possibly the pulmonary trunk, some of the main ones. The anatomy of the heart, be able to identify the different layers of the heart, various structures. Again, we're looking at another picture of the heart. Be able to tell what's right and left as far as the atriums and the ventricles. This particular picture, you have to be able to know whether you're looking at an artery or a vein or a capillary or, let's see, a gland. So once you identify it, then provide the specific anatomy of that vessel or gland as far as the outermost part, the middle part, and the innermost parts. The key here is be able to identify the upper torso arteries, know the branches, what turns into what. The next picture, what we're looking at here is the abdominal cavity. Be able to identify the arteries within the abdominal cavity. Okay, this picture here, be able to identify the upper torso veins. And once you know the arteries or the veins, you know the other. And your next picture is the identification of veins in the abdominal cavity, abdominal pelvic cavity. So know the veins in the cavity. The next picture, again, be able to identify the different anatomy of the heart. Also be able to identify the electrical system of the heart. Know where your SA nodes are, know where your AV nodes are, know where your bundle of His is, your bundle branches, your Purkinje fibers. Also, learn your coronary arteries and your coronary veins. Again, these are the arteries and veins that feed the heart itself, both anterior and posterior. Know where they drain into, or where they come from. Okay, this following picture here has got a lot of arrows pointing to it. What you're looking at is you're looking at a heart. You're looking at the various structures, the superior, inferior vena cava leading to the right atrium, which drops down into the left ventricle, or I'm sorry, the right ventricle. Then you have the left ventricle, and above it, the left atrium. We can see a pulmonary trunk. We can see an aorta, an aortic arch. We can see 3 branches coming off the aortic arch. Know what those 3 branches are that come off the aortic arch. And which one's first, second, third, etc. So this particular picture is going to ask you questions about the valves, identification of things in the heart. Okay, in this picture here, we're looking at a picture of red blood cells. Now, typically, when you see a red blood cell on a slide, you're going to see it looks like a donut. Here we see circular shape, several of those, but we also see different shapes of the red blood cell. Be able to tell me what this condition is? Okay, in this picture here, what we're seeing is the white blood cells, all of them next to each other. We've seen them prior to this individually. Or what you need to look at here is you need to be able to identify these white blood cells. 3 of them are granulated, 2 of them are not. Some are multi-lobed, the others are not. Not only identify them, but be able to explain the functions of them. Be able to explain their functions. In this picture here, we're looking at red blood cells. These particular red blood cells are clumping together, and you can see on the edges, you can see spikes. You can see spikes. Be able to tell me what this is called and the condition associated with it. Okay, in this picture here, what I'm wanting to get, or wanting you to understand from this picture here, is there is a reaction here. And understand that if there's a reaction with this, it's a positive. So we would say Rh positive. There's a positive reaction here. Be able to, or learn, what happens if a woman gives birth to a child who is Rh positive or Rh negative. So be able to explain what happens when the blood from the child crosses over to the mother. This next picture, what you're looking at is a capillary tube that was used to do a hematocrit, which we're spinning the blood down to separate the plasma from the other elements, which I've asked you to tell me what the elements are, or what the name of that is, and what's included in that. So what we're looking at here is the capillary tube. At the very right side, at the far right side, we've capped that tube with clay. Below that clay, further right, it says, well, there's a red mark on the tube. What that red mark means is that is the particular end that is heparinized. And what heparin is a blood thinner. So when you touch that end of the capillary tube to a drop of blood, it will thin the blood down and allow the blood to go up inside the capillary tube, and it uses simple air blowing across the top of the tube, and it acts like a suction to pull that blood up. So what we see here is we see elements, plasma, and how that capillary tube is utilized. In this picture here, again, be able to identify, you've seen it once, be able to identify what type of muscle this is and what the green arrow is pointing to. Also be able to tell me if this particular type of muscle is multinucleated or one nucleus, and if it's branching. Let's see the next picture, the anatomy of the heart, the base of the heart, to be able to identify some of the main vessels that you're seeing: pulmonary trunk, aortic arch, ascending aorta, descending aorta, and the various branches that come off those. Okay, this next picture, be able to identify again arteries and veins in the abdominal pelvic region. This particular picture, you've got, it's asking you to learn and identify the arteries of the lower extremities. The arteries of the lower extremities. This picture here, be able to identify what type of vessels these are or glands, and identify the anatomical structures from outside to inside. The picture below, or the next picture, identify what it is that you're seeing. Are those white blood cells, basophils, neutrophils, red blood cells? And then tell me the vessel that they're traveling in. Okay, this next picture, take-home point here is, I want you to understand that we have a tray which is a tray which is RH, and a tray which is B, but you can't see the B in the photo. But understand that there is no reaction in any of those trays. In order for it to be a reaction, you'll see clumping, which there is no clumping in these reactions. Therefore, nothing has happened. So your RH is going to be a negative because there was no reaction in that. Okay. Now, that will take care of your highlights for your lab exam. Thanks for watching, and let me double check and make sure we're okay one last time here. Yep, we're good. See you next time. Good luck. Okay.